Okay, hey guys. So what we need to do is remember a little bit about DNA structure so we can start talking about its function. And so that's the goal of this video, is just to remind ourselves a little bit about DNA structure, and then we'll transition into Sanger sequencing. So let me pull up the good old presentation, share screen. And there we go. So first of all, we're going to just remind ourselves a little bit about DNA structure. So we went over this a few weeks ago when we were talking about biomolecules, back in chapter 3. But let's just kind of remind ourselves some of the important parts. So remember that we talked about why, or like numbering carbons. And I said, that's going to become important later on when we talk about DNA replication. Well, here we are talking about DNA replication. So first thing that we need to do is, so in this particular 5 carbon sugar, we've got 1, 2, 3, 4, 5 up here. This 5 carbon sugar is deoxyribose. And so it's of DNA fame. That's what gives DNA the D part of its name, is because of this deoxyribose sugar. So first thing that we're going to do is remember how to number these carbons, because now that we need to remember how to do that so that we can talk about DNA replication. So first thing that we're going to do is find the oxygen here in our ring of our sugar. And then we're going to move in a clockwise direction. So we're going to go to this carbon. We're going to name that, I got black on my finger. We're going to name that carbon 1. We're going to call it 1 prime, and we're going to write it just 1 and then an apostrophe. Okay, so there's carbon number 1. Carbon number 2, what do you notice about carbon number 2? That's different from carbon number 3. Anything? Okay, so then we can keep going. Let's keep going. So we got carbon number 4 in this ring, and then we move out of the ring and we start talking about carbon 5. And so now when we're talking about the directionality of DNA replication, we're going from 3' to 5'. Now you guys know what that means, because we're attaching where that dehydration reaction happens between our 2 nucleotides. Hopefully these words sound familiar to you. Is happening, it's attaching this 5 carbon number 5, to carbon number 3. Okay, all right, so that's that part. If we remember back to in our discussion about monomers and polymers, the monomers of nucleic acids we call nucleotides. They come in 3 different parts. So this slide, I actually just yoinked from my biomolecule presentation and threw into this one. So hopefully this looks familiar to you. So here's our pentose sugar, our 5 carbon sugar. In this case, well, we don't know if it's DNA or RNA, because our what's on our number 2 carbon has been, it's just not there. So, okay, so that's the first part. So in our nucleotide, part number 1 is our sugar. Part number 2 is our phosphate group that carries a negative charge. That becomes important later when we start talking about DNA sequencing, talking about how DNA is hydrophilic, and that hydrophilicness comes because of this negative charge. It wants to interact with the semi-positive portion of the water molecules. Okay, so we got sugar, we got phosphate, and then our last part is our base. This varies depending on the type of nucleotide it is. So here, I think this is either an A or a T. I can't remember, because I don't have all the chemical structures memorized, and therefore I do not expect you to memorize all the chemical structures. But so I do want you to know sugar, phosphate, base, and I want you to know what carbons those things are attached to. So our base is attached to our carbon 1. Carbon 3 is an important one. Carbon 5 is an important one. Okay, that's carbon 5 is where our phosphate is grouped onto. Okay, So this all sounding familiar? We got our bases, they're A's, C's, G's, T's, or if we're talking about RNA, then we're talking about U's. So hopefully this is all coming back up to you guys, where you're remembering all this stuff that we've already talked about in this class. We've already briefly discussed Chargaff's complementary rule. That we didn't call it Chargaff's complementary rule, we just called it the complementary rule. So Chargaff is the guy that discovered this, that we see A's and T's occurring in equal proportions, and we see that C's and G's are occurring in equal portions, but those the proportionality between the pyrimidines and the primines are different, or the purines, sorry, the purines and the pyrimidines are different, but the A's and T's are always the same Z's, G and C are always the same Z's, and so that means that suggested that these are pairing up, and that's exactly what we're seeing. So and I put these hyphens in there to remind us that A's and T's make 2 hydrogen bonds between the 2 of them, and C's and G's make 3 hydrogen bonds. Okay. And so the point of that is that DNA sequences that have high amounts of C's and G's are typically more stable because there's just more bonding between them. Okay. And we remember we talked about how DNA is anti-parallel. These strands are running from the 5' to the 3' directions, and they're going in opposite directions. Okay, and I said this will become important in DNA replication later, and now here we are, it's later, so we got to talk about this. So so this is just the remembering part of this lecture. Okay, so we talked about how our 3' end and our 5' end, so there's our phosphate group, and the phosphate group of this nucleotide is attached to the 3' carbon of this nucleotide, and so forth. And then if we look at the other complementary strand, we see that it's running in the opposite direction, anti-parallel. Okay. So you guys probably are well aware that DNA forms this double helix. It's this twisted ladder shape. Spiral staircase is often a analogy that is used. It has that sugar phosphate backbone, and then the bases face the interior. And this is a discovery that was made by these dudes named Watson and Crick. And we'll talk a little bit more about them in upcoming lectures. But remember, the sugar phosphate backbones, that's kind of the up and down portions of our ladder, and then the rungs of the ladder that are going acrosswise are the bases. Twist that, and then you got a double helix. Okay. So you guys can read over the section 1 of chapter 14 that goes over the history of DNA and how it was discovered. I'm not going to spend time lecturing about it because I want to spend more time focusing on the structure of it and what it means and how it works and what it means for living organisms. But I also put a link into this crash course biology video, a little bit about how DNA was discovered. It's a really good video, and they cover Rosalind Franklin really nicely, which most of the time when we talk about the discovery of DNA, she's kind of like, oh yeah, that was really unfortunate, but this she's the main focus in this video, which I appreciate. Okay, end of rant. The takeaways are that we have semi-conservative replication, which means that when we replicate DNA, one of those parental strains acts as the template for the new strain. So we unwind that DNA, we separate it, we unzip those 2 different DNA strands that form the double helix. Now we have 2 single strands, and then we can create a new copy as indicated by these kind of these bubble letters in this little cartoon here. This is another book that I find really interesting. I have a hard copy of it right over here. This is really fun. It makes learning all of this stuff a lot easier because it's in like comic book form instead of like boring textbook form. So there's a link to the PDF version of this book. And when we talk about Punnett squares, we'll pull this out and use this because I find it's actually a lot more helpful and a lot more engaging to look at it in this format. All right, end of rant. Boom. Okay, so I put these in here. You guys have links to these slides, so you can use these to practice pairing. And if you're not sure what that means, so what we would do is we would say this T goes to an A, this T goes to an A, this T goes to a G, this C, or sorry, this C goes to a G, this C goes to a G, blah, 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 blah. So practice using Chargaff's rules to match these base pairs. Let me know if you have any questions or concerns about that, because this was going to be important later when we talk about DNA replication, and then we're going to talk about transcription and translation. And then when we're going from DNA to RNA, we want to know these rules down packed. Okay, so practice that. Okay, so now we can start talking about how we can use the DNA structure to actually obtain DNA sequences, which can then allow us to ask ecological and evolutionary population level genetics questions in science that we weren't able to have prior to 1977. Okay, let's get into that. So basically what we want to know from this section of the textbook is the structure of DNA. We just covered that, so I'm not going to do it again. We're going to talk about how Fred Sanger, pictured here, lovely old Freddie, won his second Nobel Prize by figuring out how to sequence DNA. It's very similar to the way that he figured out how to sequence amino acids and proteins to obtain the sequence for proteins. And then we'll talk a little bit about prokaryotic and eukaryotic DNA. Well, they're very similar. Okay, so prior to 1953, we didn't know the structure of DNA. We knew the components. We knew that it was a sugar. We knew that it was a 5-carbon sugar. We knew that there was phosphate involved. We knew that there were nitrogenous bases, but we didn't know how the pieces fit together. And so these two dudes, Pauling and Corey, suggested that there is a triple helix. There's 3 strands of DNA where the bases are facing toward the outside and the sugar phosphate is on the inside. And so here's another little YouTube video that talks a little bit about the DNA structure and the discovery that was made by Watson and Crick about the structure of DNA. That's a useful study tool if you're finding that you need a little bit extra practice with this stuff. Okay, so, but what happened was that James Watson and Francis Crick literally stole off of Rosalind Franklin's desk these X-ray images that showed that DNA forms a double helix. And so that's, so Franklin's data showed that no, it can't be a triple helix, it has to be a double helix. And so that was kind of the missing piece, and knowing that information allowed Francis Crick and James Watson to be able to figure out, oh yeah, the phosphate sugar is on the outside, the bases are facing inside. That jives with the Chargaff's rules about the base pairing. And so they were able to actually, by literally stealing Rosalind Franklin's data, they were able to be the first to publish this structure in a Nature, in the scientific journal Nature, back in 1953. What was really sad is that Watson and Crick and Rosalind Franklin's PhD advisor were awarded the Nobel Prize for making this discovery in 1962. But at that time, Rosalind Franklin had already passed away at the age of 37 because of ovarian cancer that she probably got by creating these images, these X-ray images. She was exposed to high amounts of radiation by doing the work that she never got credit for. And so that's a really sad story, and it's probably one of the best documented cases of sexism in science. So that's a bummer, but we're going to keep moving on. Okay, so Watson and Crick said, hey, we found this structure, it's really cool, and I think that we understand how DNA can replicate because now we understand the structure. And so like, here's the direct quote, you guys are welcome to read that, but that's exactly what happened, is that understanding how DNA is put together, we can also understand how it is copied. And so this semi-conservative replication, where we have the parent strand of the DNA is unwound and denatured. So that means it's melted into 2 single strands, and then we use one of those single strands as a template to make a copy. And the same thing is happening with the other one. And now we have two copies of the DNA molecules. One of the strands is from the original, the OG strand, and then one is the newly formed. Okay, and so this was discovered using isotope labels, and we'll go into the experiment a little bit more detail later, but also know that, hey, isotopes, remember we talked about those and we were like, who cares about isotopes? Well, if you want to understand DNA replication, you do. Okay, so using these same ideas of how DNA is replicated, we can actually, what Fred Sanger did was figure out how we can use that replication to actually give us the DNA sequence and the order of those base pairs. And so then that we can use that information to understand all these other kinds of questions that were previously inaccessible to us. And so basically what Fred Sanger does is takes that DNA, breaks it down, and then sorts it out by size. So we'll get into all that. So first of all, we need to understand the ingredients to this reaction before we can talk about how we actually get the things. So whenever we're doing DNA replication, we have DNTPs. Okay, so the D stands for deoxyribose, right? That's just our regular old our sugar that makes up DNA. The N is the variable. So it's just like, in algebra, we talk about the X being the variable, and then we have to find X. So in this case, N is the variable. So we can have C, D, C, TPs, DA, NT, or TPs. Saying NTPs is like kind of muscle memory for me. But so that's where these letters, the bases come in, is plopping those into where the N is. T stands for tri. So here, instead of in our regular old nucleotides, we only have one phosphorus group. But as we're doing DNA replication, whether that's artificially while we do Sanger sequencing or naturally happening in your own body right exactly now, we have a triphosphate group on our sugars. Okay, so that is going to provide the energy and the nucleotide. So here's our nucleotide. It's just one phosphate, our sugar and our base. But here we have these 2 extra little energy storage molecules or groups happening to be right there. So that's super convenient that we have our building blocks and an energy source. Okay, so P stands for phosphate. So that's what the DNTP is, deoxy, variable name for our nitrous bases, T for tri, P for phosphate. Okay, and then in DNA, in Sanger sequencing, each of these bases has like a little fluorescent dye that's attached to it that's unique to each of the bases. So when we shoot a laser at it, we can tell what base it is by the color that it lights up as. Okay, so we also need to add DDNTPs. what is a DDNTP? Well, remember that D stands for deoxy. So that means in this DNTP that we have here, we have on our second carbon, our 2' carbon, we are missing an oxygen atom. Okay, but in our DDNTPs, that's our dideoxy, all that stuff. That means that on our 3' carbon, 3', remember 3' was important. We're missing an oxygen atom on our 3' end. Why do we care that we don't have an oxygen atom right there? Well, if we look back at how these nucleotides hook together, that phosphate group on our 5' end has to hook to the oxygen atom on our 3' end. If we don't have an oxygen on that 3' end, that means that boom, that's where DNA sequencing ends. Okay. So what we got to do in order to have this dideoxy chain termination method, which is a fancy name for Sanger sequencing. Sanger sequencing is easier to say, so that's how we talk about it. And also props to Freddie who figured this out. So here's what we do. We got to extract the DNA. We got to make a lot of copies of this DNA using a method called PCR, which is called polymerase chain reaction. We'll talk about that later. But that allows us to make copies of our certain portion of the DNA that we care about. And then we got to take our double-stranded DNA and melt it into 2 single strands. Then we got to pump a bunch of DNTPs and a bunch of DDNTPs into our mix. And those are incorporated onto our template strand. Boop, 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 boop. Once we get a DDNTP, then that particular sequence is terminated. And so now what we'll do is we end up with this pool of all these different sizes of DNA, and we can sort those out using this method called electrophoresis, which is basically like little molecules move faster and big molecules move slow. So it's like a little DNA race. And then what happens in the DNA sequencing machine is that there's a little laser that zaps them, and then the fluorescent dyes light up, and then a computer will record the order of those fluorescent flashes. And that gives us the DNA sequence. Okay, let's go through these steps a little bit easier, like a little bit more slowly so we can understand them. So DNA extractions, we are going to actually do this tomorrow on Tuesday. So make sure that you read the protocol before you come to class and make sure that everything goes nice and smooth. That's on Canvas already for you. Okay, then we gotta do this other thing called PCR. And we don't have to do this, it just makes it a lot easier by increasing the number of copies that we have at our disposal. And it also gets rid of all the DNA that we don't care about in this specific gene region. It makes copies of that region, and like all the rest of the DNA from our organism that we don't actually care about is not copied. And so that it just makes it sequencing much easier. And then this PCR process was invented or discovered by this guy named Kary Mullis, who is wacky, to say the least. He's a super smart dude, and there might have been some drugs involved. All right. And so here is one of my high school kids. This is Robin. She was a student that I worked with about 3 years ago now, I guess. So now she's at what's the one in Tucson? She's at the University in Tucson, doing research as an undergraduate. So that's pretty cool. All right. So we got to denature the DNA. That just means that we're going to add heat, and that's going to that increased vibrations of those molecules or those atoms are going to break those hydrogen bonds and release the hydrogen bonding that's keeping those bases together. So we actually end up with 2 single strands. And that's what we mean by denaturing DNA or melting it. It's the same thing, just unwinding it. And then we have DNA replication happening from a pool of dNTPs and ddNTPs. Remember, these are the nucleotides that have been modified slightly, and the ddNTPs lack that 3' oxygen. Okay. And so what happens is that we end up with all of these DNA sequences of various sizes kind of floating around in our little tiny tube of clear fluid. And so what we need to do then is run it through this. So this is what our Sanger sequencing machines look like. And this is the one that I've used, or a machine like this. This isn't the exact same one, but it looks exactly like this. And what happens when you open up this door, it looks like this, and you have your 96 well plates. So you have 96 samples, and there's these little metal pegs that sit down inside of each sample, these little capillaries, these little teeny tiny tubes that suck up all the goo that we have in those samples, and it runs it through this copper wire. And so the DNA is actually transported through these copper capillary tubes. And what we see is that our small molecules will move really, really fast. And then our big molecules are like bumping into stuff, and they're like, and it's harder. It takes more time for them to transport all the way through all of this copper capillary tube action to get zapped by the laser. And so, and then we have there's a computer that's attached to the Sanger sequencer that's recording all of the fluorescent flashes from those dyes. Okay, yeah, and then I just talk about that. So the laser that's actually, I'm gonna change that word now. In this photo, in my machine that I use at NAU, it's up there, but this one looks like it's down here. Okay. So that's where that laser is. And then it translates those flashes of color into an actual DNA sequence. Okay, so how do we actually, so this method was invented in 1977. It was the primary method of obtaining the human genome that took place in the 90s, and I think it took from 1992, I can't remember, but it was finished. It took about 11 years to completely sequence the entire human genome, and they use Sanger sequencing for this. So what we do in my lab at NAU is employ this method in ecological questions. And I think the easiest one to explain, and the one that is the most meaningful, is this concept, this weird system out at Sunset Crater that is just about 20 or 30 minutes on 89A heading out of town. So what you'll see on Sunset Crater, like, first of all, this is a volcano that exploded about 1,000 years ago. And you can see that the soil here doesn't really look like soil very much. It's kind of like just black cinders. And it gets really hot there, and it's really dry. And that the cinders don't hold on to the water. It just goes straight through. So this is a really harsh environment for these plants to try to live there. And what we found, or like, back in the 80s, when Dr. Tom Whittam, who has just retired from NAU, when he first started his career, he would take students to these field sites and he would say, okay, go wander around and look at stuff and find a cool question and come and then design a study and answer your question. And it was this really great, like, boot camp scientific method way to do ecology. But what one of these early students found is that they were like, okay, here's a pinyon pine that looks like a regular pinyon pine tree. That's normal. But here's a pinyon pine that looks like it's being grazed, or it's constantly being hedged. You guys have probably seen those English gardens that are like heavily pruned, and there's all these topiaries and these like tree sculpture things. It's kind of what this tree looks like. And so here's me for scale, and you can see that it, like, What is up with that? Why do these trees that are growing next to each other look so different? And through decades of study, we've actually discovered that there's a moth that attacks these particular trees that have these kind of weird wonky shapes and that don't really attack the ones that have the regular shapes. Okay, so then the question is why? Well, one of the reasons why is because these particular trees that look like normal use the bugs will actually attack them, but the trees will defend themselves by pushing out that moth, that little caterpillar that bores into the tips of the tree, pushes them out using a bunch of sap. And so then that sap means that those trees need to use a lot of water. These trees, the wonky trees, don't do that. They just allow the bugs to eat them. And then they actually are more drought tolerant. So in 2002, when our big drought that we're still in hit, they're like, oh, dude, these little wonky trees, these guys are kind of screwed. And what they found was actually that it was the opposite pattern. It was the normal trees that used a lot of water that had about 70-something% mortality during this drought, whereas these wonky looking dudes only had about 20%. And so it was very different from what these researchers expected. And now bringing it back to Sanger sequencing, one of the things that my advisor right here, Kitty Gehring, is interested in is plant-associated fungi, specifically mycorrhizal fungi. So these are fungi that form symbiotic relationships with the roots of most plant species, including pinyon pines. Pines have to have their little mycorrhizal buddies. So we can see a root here, and all this white fluff is the fungi that's living with the root. And the fungus provides water and nutrients to the plant provides the fungus sugar it makes through photosynthesis. And so now both parties are benefiting from each other. What's interesting is that there's lots of different species of fungi that can form these types of relationships. And what they saw is that on these wonky trees, they had the species of fungi that provided the best drought tolerance benefits to the tree, whereas these tall upright guys didn't have those, had a different group of species that associated with the roots that didn't convey this drought tolerance. Now, if we circle all the way back to why it's interesting that chloroplasts, remember those little organelles? Chloroplasts in pines are inherited from their dads. And so now we can actually use Sanger sequencing to sequence the DNA inside the chloroplasts to know who the dads of these trees are. We know who the moms are because we collected the seeds from the moms. And now we can know both parentages. And what we find is actually that these wonky trees tend to be the dads of a lot of the babies. And so not only knowing what fungi are associated with the roots, but also knowing their parentage, their heritage, allows we can answer both of those very different types of questions using Sanger sequencing. Okay, end of rant. All right. So the way that DNA is packed in prokaryotes and eukaryotes is obviously going to be very different. So what we find, again, if we just look at the structure, that we know that eukaryotes have a true nucleus. That's what their name means. And that's where their DNA is kept, is in the nucleus. And then in prokaryotes, we have the nucleoid, which is kind of this, a portion of the prokaryote where we actually see the chromosomes. And remember, in prokaryotes, it's one circular chromosome. In eukaryotes, it's lots of linear chromosomes. Okay, so prokaryote organisms have a nucleoid region. So typically in our double helix, as we're thinking about how it gives us turn, there's 10, so like in one of these little twists, there's 10 bases. But what we see in our, we get my visual aid. What we see in our prokaryotes is if we take the circular, let's pretend that this hairband, oh my God, I just lost another one. Let's pretend that this hairband is a chromosome of a prokaryote. We can actually take this thing and we can make it take up less space by coiling it up and bunching it up. And now it takes up very little space, right? So that means that we're have supercoiling. So that means it's either overwound or underwound. And then it requires, because like if I let go of that hairband, it's going to go back to its circular shape. So it requires enzymes and other proteins to kind of keep it in this bunched up form in the nucleoid region. Okay, but then if we talk about eukaryotes, again, this is stuff that we've already discussed a little bit, so hopefully this sounds familiar. So we have these linear structures, that's, you know, our DNA, our double helix up here at the very tippity-toppity of this figure. And then the DNA will wrap around these special proteins called histones. And then once they get all this nice little, it creates this name that we call a nucleosome. So this is a body of, you know, nuclear stuff. And so then that's kind of gives us this beads on a string idea where we have all these histone proteins that are attached by these little linker DNA segments. And so then we get this more and more and more packing when the cell's ready to duplicate and it needs to provide the genes for the daughter cell. It starts to get this more and more condensed chromatin structure, and eventually it gets to be a thing that we can see using microscopes and stuff as a chromosome. Okay, so I will end it there for today. So that's a lot for you guys to unpack, and we will practice Sanger sequencing on tomorrow after we do the DNA extractions. Okay, see you tomorrow.